Thank you for joining us today and allowing us to share with you the latest development on the MARS technology. The MARS system is a multiphysics-based approach that allows for automated sample preparation as well as cell isolation. The system consists of 3 modules: the cell washing module, cell concentration module, as well as an immunomagnetic module. This process allows you to give a one-step approach to taking a sample and allowing it to go through the process and getting the target cells of interest in a very fast automated process. The throughput is also a very high throughput at 1 ml per minute flow rate. The multiphysics-based approach is configured with 2 core technology: the acoustics-based technology that allows us to do cell washing and concentration. Using acoustics or sound waves, we actually move cells to the center stream while maintaining molecular debris, red blood cell, and platelet debris in the outer stream. This allows us to collect a purified population of white blood cells in our MARS CPC chip, the cell processing chip. From there, we can then move the cells to an immunomagnetic separation process that allows us to label the target cells of interest or the non-target cells of interest and magnetically separate, purify them in a single step without the use of a matrix. Our column does not contain a matrix, which minimizes cell loss, giving us not only high purity, but high recovery of the target cells. How does our acoustic technology work? Here's an example showing beads in both large and small sized beads. From 3 to 5 microns, these large beads can be actually moved and collected in the center stream while removing the smaller objects, such as represented by this 1 micron bead particle. This allows us for a very quick, clean process of obtaining target cells based on their size and density while removing smaller objects and maintaining them in the debris stream. An example of where we've applied this technology is being able to actually cleanly process your sample. Here we have lysed whole blood that's processed through our CPC chip, and what we get out is basically a purified collection of white blood cells, while in the debris stream, we remove the red blood cells, platelets, and molecular debris. As you can see here represented in the bar, the original sample of lysed no wash in the red bar represents the MARS wash sample, and the blue represents the centrifuge wash results. And you can see through our MARS wash chip, we recover the majority of the white blood cells, lymphocytes, monocytes, and granulocytes. Not only that, but within the subset population of the lymphocytes, we can see that T, B, and NK cells are also highly recovered. Recovery of the white blood cell is a core objective. However, removal of the molecular debris is also a high objective using the MARS CPC chip. Here we can see a sample of the original blood. Using flow cytometry, we can see that there's a significant amount of debris that is in the sample compared to the white blood cell population labeled by CD45. In centrifuge wash, while it does remove a large portion of the debris, the MARS wash shows significant removal of the molecular debris without compromising the loss of white blood cells. We use this application in order to purify white blood cells from whole blood, bone marrow, apheresis sample, frozen PBMCs, as well as dissociated tumor samples. In another application of our technology, rare cell isolation, we have focused on the minimum residual disease solution in partnership with the University of Pennsylvania. Current methods today show a lack of sensitivity. At time of diagnosis, we can see a significant number of tumor cells. After treatment, there are very few residual cells remaining. And it's important to be able to monitor these MRDs for treatment and remission. But today's methods lacks the sensitivity. By flow cytometry, NGS, and PCR, being able to achieve up to 1 in a million cell sensitivity is difficult. A good enrichment requires greater than 1 in a million cells, not only in the sensitivity, but also in an automated, fast throughput manner. Our Mars technology was applied at the University of Pennsylvania Cancer Biology Center in Dr. Louis Chodosh's lab, as represented by the work of Dr. Elizabeth Chislock. She was able to show that using our Mars technology, we were able to compress the processing time by at least 4-folds, as represented here. What typically would take all day to be able to process a sample, she was able to do in less than 1.5 hours, with a lot fewer hands-on manipulation time. Time compression is one thing, but can we achieve greater sensitivity? And the results in the preliminary test that Dr. Chislock has shown here, that tumor cells were typically recovered in excess of 80%. And not only that, but down as low as 1 in 5 million bone marrow cell frequency. With these results, Dr. Chislock and Dr. Chodosh's lab is now moving that into a multi-center clinical trial. In another application, we'll show the immune cell isolation, both positive and negative cell selection of T cells. The first process will show you the negative enrichment of these T cells. And we begin by actually taking whole blood labeled with the target antibodies of interest. In this particular experiment, we used CD14, CD15, CD19, and CD56 to isolate cells of non-interest into the positive fraction while collecting the T cells, the remaining T cells, in the negative fraction. So once the cells have been labeled, lysis buffer is then added and placed onto the system. The system is in a one-step process, can take the cells and be able to wash the cells, concentrate them, and at that point, if you are using a non-indirect method, in this particular experiment, we used biotinylated antibody to start and used our streptavidin magnetic beads as a labeling step. These beads were then added and then post-incubation processed through the magnetic separation module. The results of that is shown here. In CD3, the CD3 population is shown by the red population as noted here using CD3 FITC. And this is before isolation. And in the negative fraction where we expect our T cells to be, you can see that we have a large enrichment of the T cells representing 91% purity and approximately 95% recovery of the T cells of interest, while the cells of non-interest are in the positive fraction, which was magnetically pulled out. In another process, we wanted to show the positive isolation of immune cells using a no-lyse, no-wash sample process. This saves a significant amount of time as well as reduction in loss of cells. So as you can see here in the Mars processing step, we take the cells, magnetically label them, and then put them immediately onto the cell separation module. This takes less than 30 minutes to be able to process the sample. In conventional methods, it could take up to 2 hours or more in order to process the sample for isolation. The results of that are shown here. These are CD3 T cells isolated from an apheresis product. The cells we began with are 267 million white blood cells, and in the positive fraction, we were able to isolate 116 million CD3 positive cells. And the purity of those cells are shown here to be at 96%. In the negative fraction, we can see very few T cells were lost, and the majority of the cells that were there were B cells, along with other cell types. Not only are we able to process a large amount of cell sample, over 100 million CD3 positive cells in a single column run, we were able to get very high recovery that's greater than 95% purity and greater than 95% recovery. We then took those cells and cultured them by splitting the cells in half. Half were unstimulated, The other half were stimulated using a bead-based stimulation of CD3 and CD28 without IL-2. As after day 3, we were able to show CD25 positive cells were the majority of the cells, showing that the cells are activated and beginning their process of proliferation. And that the results of that is, as you can see here, the unstimulated cells did not grow in culture. They began to diminish in culture, but the red represents the activated cells. And after 7 days, we were able to show more than an 8-fold expansion of the T cells. Not only did the T cells expand, but as we can see here, they are the majority of the cells are central memory T cells, as shown by CD45 RA negative, CD62 L positive. In summary, the positive and negative selection of T cells were achieved with greater than 90% purity and recovery. Cell selection can be achieved in under 30 minutes. As part of that process, we also obtain high viability of these cells with minimal processing time as well as minimal manipulations. Greater than 100 million cells were isolated in a single run in a single column. T cells isolated and cultured show high proliferation of over 8-folds within 7 days. And the majority of those T cells were central memory cells as desired in immunotherapeutic. A variety of cell types are compatible with the Mars system: whole blood, bone marrow, apheresis, frozen PBMCs, and dissociated tissue samples. Now that we've shown you the Mars product, I'd like to introduce you to the latest member of our Mars family, the Mars bar, to address the higher capacity and larger volume capability. Our Mars sample prep system and our Mars cell separation has now been given a high capacity solution with the Mars bar. To learn more about our Mars product and the Mars Bar Early Access Program, please contact us at info@appliedcells.com. Thank you.